Great, David, thank you very much, and thank you for presenting that so quickly. Very, very interesting and exciting stuff. Our next presenting company is TiGenix, and I'd like to ask Eduardo Bravo, the CEO, to come and tell us the story.
Hi, thank you for being here. And if I need to talk as fast, I will have to do it in Spanish, so I will do my best, but I'm not sure that I will get there. So maybe I'm experiencing the same trouble, so I don't know why. Thanks. So we're a public company. I'm going to be making forward-looking statements. And we are a quoted European cell therapy company based in Belgium. We have the first ever cell therapy product approved in Europe. That was in 2009. And after that, we have a platform of allogeneic adipose-derived stem cells products. We do not have the blue chip investors that the previous presenter was saying, but we are probably the only company that has 30% of the stock in the hands of very well-known pharma companies. Grifols, Roche, and Novartis own 30% of the stock of TiGenix. And we have about $40 million of cash available. This is our pipeline with an autologous product, ChondroSelect, already in the market in Europe, and then a platform of local, intravenous, and intralymphatic administration of adipose-derived stem cells for the treatment of inflammatory and autoimmune diseases, and I will go into the details of those products during the presentation. We just presented a couple of weeks ago the full year results for 2013. Everything is moving in the right direction. I think that the key points there was that we finally got confirmation of our regulatory pathway for 601 in the US. We are well underway on the recruitment of the pivotal phase 3 trial in Europe, and the 18 million investment of Grifols that acquired 20% of TiGenix strategic investors and helped us in raising additional capital towards the end of the year. Let me spend a few minutes on 601, which is our leading asset. This is a local injection of the cells for complex perianal fistula in Crohn's patients. I'm not going to spend time on Crohn's. Everyone knows Crohn's. Something that people do not realize is that about 1 in 10 patients that suffer from Crohn's will end up suffering from those chronic perianal fistulas. And those chronic perianal fistulas, despite the problems for the patients today, do not have a cure. Infliximab is the only approved product for fistulizing Crohn, despite a very low efficacy and very high recurrence rates. The only other option will be surgery, but in about 1/3 of the patients you end up with anal incontinence. I also have some very nice videos, but I'm going to spare you the videos of the indication here. So what do we offer here is 601, which is a very simple injection of these adipose-derived stem cells in the fistula walls to promote suppression of inflammation and natural healing to happen. And again, as I mentioned before, we're in the middle of a single pivotal phase 3 in Europe after having done some significant research in patients in Europe, and we expect to file in 2016. Again, not being a scientist, I'm not going to go through the mechanism of action of the cells. I think that there's enough companies today that explain that for mesenchymal stem cells in general is those paracrine effects that produce this very strong suppression of inflammation and reduction of the pro-inflammatory cytokines and increment or induction of anti-inflammatory cytokines. That's the key mechanism of action of the cells. We are one of the very few companies in the world working with adipose as a source of those stem cells. And there's more and more evidence that shows that these adipose-derived stem cells may have some advantages, at least in vitro. The most important ones that have to do with the yield, have to do with the stability and expansion, and probably the most important one is the lower immunogenicity, at least in vitro. In our platform, we've been 6 times in front of the regulatory agency to ensure that our preclinical package and our CMC package was completely validated. We have a GMP manufacturing facility that is fully approved, and as I mentioned, after 6 times in front of the EMA, the clinical development pathway has also been clear with the authorities. We've done significant clinical experience with the product. We had an autologous product first that did all the development with efficacies always above 40%. We then did a small phase 2 trial with the allogeneic follow-up, an investigator-initiated trial in rectovaginal fistulas, and actually there's been now a Korean company that has done a phase 2 clinical trial that has demonstrated even higher efficacy. So it's good to know that these cells work in this indication even on someone else's hands. Just to give you an idea how tough this patient population can be and what's the effect of the cells, this is the phase 2 clinical trial, which was a randomized clinical trial in 50 patients, and in patients that had 80% of the patients had previous surgery, 20% already anal incontinence, 60% suprasphincteric fistulas. Well, in those very, very difficult to treat patient populations, our cells combined with fibrin glue were capable of closing 71% of the fistulas. And we're talking about curing the fistulas, complete closure of the fistula tract, including repitalization of the external opening. Not only 71% close short term, but close to 60% of the fistulas remain closed 1 year after the injection of the cells, showing that natural healing really happens. We've gone into the allogeneic follow-up, again demonstrating similar efficacy on Crohn patients to whom we took away the underlying treatment for their Crohn, so the anti-TNFs were forbidden, and despite that, we were able to close 6 months after the injection more than 50% of the fistulas that were treated. And on those patients, this was the first time we were administering these cells from an allogeneic origin. We demonstrated that there was no immune reaction and that safety was extremely high. There's been also a small investigator-initiated trial in rectovaginal fistulas. Those are extremely tough to treat fistulas. Those were 10 women that had gone through several previous interventions trying to do a flap to cover the fistula without success. Well, 7 patients completed the 1 year. 4 of those patients had their fistulas completely closed 1 year after treatment. So again, very significant efficacy of the cells in this very tough to treat patient population. So with those results, we went to the EMA. We agreed on a design for a single pivotal phase 3 trial that, if positive, will allow us to file for marketing approval. Primary endpoint will remain as clinical remission, so we're looking at curing the fistula in these Crohn patients. Again, today, we've opened the 8 countries, more than 50 centers. We've recruited over 2/3 of the patients that are needed. We're anticipating to finalize the recruitment this year with data in the summer of next year. So we are basically a little bit over a year away from pivotal phase 3 data that may allow us to file in Europe. Very importantly, as I mentioned, we've gone for an end of phase 2 meeting with the FDA, and the FDA have confirmed, A, that we don't need to do any further preclinical work, that the European trial will be considered one of the 2 pivotal trials, and that we only need to do another 200 patients clinical trial in the US to file for a BLA. So we're basically 1 trial away as well to file for a BLA in the US. Finally, let me spend 2 minutes on our allogeneic systemic delivery platform, and we're targeting here the autoimmune disease space. Let me show very quickly that we have several animal models in which we've demonstrated the effect. This is the chicken-induced arthritis model. The cells compared with a vehicle. We've compared the cells with methotrexate and anti-TNF, showing comparable efficacy of the cells to these very used agents. We've demonstrated the effect of the cells in vitro in human cells of RA patients. We've demonstrated the effect of the cells, very significant, with several routes of administration in a very well-known IBD model, which is the DSS model. And we've demonstrated, probably the most impressive of all, that in 2 sepsis models, on a dose-dependent manner, we can even reduce mortality of the mouse. But that's in animals, and as you know, not always the animal translates. There is clearly a need for new mechanism of actions in these severe inflammatory and autoimmune diseases. Side effects are very strong of the very effective therapies available, and something very important is about 20% of the patients do not respond. So what we decided is to take our compound, and we did the first ever randomized controlled clinical trial in RA as a proof of concept. And we went for a phase 2A blinded trial comparing our cells with methotrexate, again, methotrexate plus placebo in 53 patients. Very tough patient population. We ended up with patients after 13 years of treatment, 3 non-biological DMARDs, followed by 3 biological DMARDs. So basically, everything that you can throw those patients had been thrown at those patients without success. Of course, primary endpoint, that was the first trial ever, was safety, extremely nice safety and very favorable, of course, if you compare it with the safety profile of the biologics. But I think that the most impressive data was actually the efficacy. And in those almost impossible patients, after everything has failed, 39% of the patients were still good to moderate responders 3 months after receiving just 3 doses of 611, and 24, 6 months after treatment. Even for me, more impressively, is that if you look at remission, so remission after 13 years, 10% of the patients were on remission 6 months after receiving just 3 doses of 611. So that was so impressive that was selected for a plenary session at the American College of Rheumatology, and we presented the data in October this year. So today we are working with probably the biggest key opinion leaders in the world in 3 different areas: RA, sepsis, and lupus nephritis, and we've agreed that we will present a full development plan probably in the month of May, deciding which of the indications we're going to take forward for future development. So in summary, you know, we have an approved product. We have a phase 3 asset going to be filed in 2016 and going to deliver data in 2015, a US pathway to the US clear, and positive phase 2 data in a very large autoimmune indication. And with that and the milestones for next year, I will leave it here, and we'll be happy to take questions outside if needed. Thank you.